Welcome to today's lesson on the testing and trialing of new medical drugs. In this session, we will be exploring the crucial steps that are taken to ensure that new drugs are both safe and effective before they can be used to treat patients. In our lesson today, we'll look at the different phases of drug testing and clinical trials, the importance of each phase, and the ethical considerations involved at each. By understanding these steps, you'll gain an insight into the rigorous processes that protect us from harmful and ineffective medications. So far, you have learned about the origin of some significant drugs such as aspirin, digitalis, and penicillin, which were developed from plants and microorganisms. You've also learned about the generalities of modern methods of developing drugs, including identifying potential compounds, preclinical testing, and modifying natural compounds in order to enhance their therapeutic properties. Building on this, today, we will delve into how new drugs, regardless of their origin, undergo stringent testing. This understanding is vital because it shows how scientific research and stringent protocols ensure the safety and effectiveness of new treatments, thus advancing our capacity to combat various diseases and improve healthcare. To understand how new medical drugs are tested and approved, we need to delve into the meticulous and multi-stage process involved. This ensures that any new medication is both safe and effective for human use. Let's break down this process. Before any new drug is tested on living organisms, it is first evaluated in the laboratory using human cells and tissues. This initial step helps researchers to determine two critical factors. The first is toxicity. Does the drug have any harmful effects on the cells? The second is efficacy. Does the drug produce the desired effect? By using only cells and tissues, scientists can get a preliminary indication of how the drug interacts with human biology, without posing any risk to living creatures. However, this method cannot provide a complete picture of the drug's impact on an entire organism. Next, the drug is tested on live animals. This stage is crucial because it provides insights into how the drug affects an entire living organism, something that can't be fully evaluated by only testing on cells and tissues. In Britain, the law requires that any new drug is tested on at least 2 different species of live mammals. This stage addresses several key questions. We need to know if the drug is still effective when administered to a whole organism, any potential toxic effects, and the right dosage that will achieve the desired effect. While testing on animals is controversial, most believe it is a necessary step to ensure that new drugs are safe to administer, before the step is taken to give them to actual humans. The final stage of drug development involves testing the drug on human volunteers through a series of clinical trials. This is a multi-phase process. Initially, the drug is given to a small group of healthy volunteers. This is to ensure that the drug does not cause any unexpected adverse reactions when the body is functioning normally. The doses start very low and are gradually increased. If the initial tests show the drug to be safe, then subsequent trials involve patients who have the condition the drug aims to treat. This helps to determine the optimal dose, which is the dose that is most effective with the fewest side effects, and the drug's efficacy when treating the condition. To ensure objective results, patients are randomly divided into 2 groups. One group receives the new drug, while the other receives a placebo, which is an inactive substance with no therapeutic effect. This setup helps account for the placebo effect, where patients feel better simply because they believe they are receiving treatment. In some clinical trials, neither the patients nor the doctors know who is receiving the drug and who is receiving the placebo. This method is known as a double-blind study and helps to remove bias from the results. Before the results of any drug trial are published, they must undergo peer review. This means that other experts in the field evaluate the study to ensure that it is accurate and that its conclusions are valid. Peer review helps to prevent false claims, ensure that the research methodology is sound, and to confirm that any findings are reliable. Today's lesson highlighted the importance of testing and trialing new medical drugs to ensure their safety and effectiveness. We began with the drug discovery phase, which involves identifying potential compounds and conducting lab research on cells and tissues to assess their mechanisms and benefits. Next, we covered preclinical testing on animals to evaluate safety and efficacy, ultimately determining if the drug should proceed to human trials. We then discussed the phases of clinical trials on groups that increase in size. This includes the use of placebos in double-blind trials that prevent biases arising from the placebo effect. Finally, we addressed regulatory approval, where authorities review clinical trial data to ensure that the drug meets our strict safety and efficacy standards. Understanding this process reveals the rigorous journey every drug must take before it reaches human patients.